Hey everyone! Tonight we're exploring the fascinating world of bioluminescent creatures and their remarkable ability to create light in the darkness. Imagine having your own personal nightlight built right into your body, powered by nothing more than chemistry and evolution. From the depths of the ocean to your own backyard, these living lanterns have developed some of nature's most enchanting adaptations, and nature has crafted solutions we're only beginning to understand. So, before you get comfortable, take a moment to like the video and subscribe, but only if you genuinely enjoy what I do here. I'd love to hear in the comments which bioluminescent fact surprises you most tonight. Now, dim the lights, maybe turn on a fan for that soft background hum, and let's ease into tonight's journey through nature's remarkable designs. In meadows and forests around the world, as twilight deepens into night, tiny alchemists begin their ancient work. Fireflies, those beloved beetles of summer evenings, carry within their abdomens one of nature's most elegant chemical reactions. Scientists call it the luciferase reaction, a process so efficient that it converts nearly 100% of its energy into light, producing almost no heat. Compare this to an incandescent bulb, which wastes 90% of its energy as heat, and you begin to appreciate the sophistication of this six-legged lantern. The chemistry unfolds like a perfectly choreographed dance. Inside special cells called photocytes, the firefly combines luciferin, a light-emitting compound, with the enzyme luciferase. Add a dash of oxygen, some ATP for energy, and magnesium ions as catalysts, and suddenly cold light blooms in the darkness. The firefly controls this reaction with remarkable precision, regulating oxygen flow to create the characteristic flashing patterns we see floating through summer air. Did you know that each firefly species has its own unique flash pattern? Like Morse code written in light, these patterns serve as identification cards in the darkness. The common eastern firefly, Photinus pyralis, produces a distinctive J-shaped swoop of light, rising upward for about half a second. Meanwhile, their cousins, the synchronous fireflies of the Great Smoky Mountains, have mastered something even more extraordinary. Thousands of males flash in perfect unison, creating waves of light that ripple across entire mountainsides, a phenomenon so rare it occurs in only a handful of places worldwide. Research reveals that fireflies can adjust the color of their light by tweaking the chemistry slightly. Most North American species produce yellow-green light at a wavelength of about 560 nanometers, the exact color to which firefly eyes are most sensitive. But some tropical species push into orange or even blue territories, each shade perfectly tuned to their environment and needs. The efficiency of this system continues to inspire scientists developing new forms of sustainable lighting, from glow-in-the-dark trees to bioluminescent street lamps. The gentle pulse of firefly light serves multiple purposes beyond romance. Young firefly larvae, glowing steadily in the leaf litter, advertise their toxicity to predators. These glowworms, as they're sometimes called, contain defensive chemicals that make them thoroughly unpalatable. Their constant glow says, quite simply, "I'm not worth eating." Even firefly eggs sometimes glow softly, nature's own nightlight protecting the next generation. Scientists recently discovered that some fireflies can synchronize their flashing using a phenomenon similar to how human hearts can sync to music. Each firefly acts as a tiny oscillator, and when males gather in groups, they naturally begin to flash together, creating one of nature's most mesmerizing displays. The precision required for this synchronization rivals that of any human orchestra, yet it emerges spontaneously from simple chemical reactions and behavioral rules. In Japanese culture, firefly viewing, or hotarugari, has been cherished for over 1,000 years. Ancient poets wrote of fireflies as the souls of warriors, their lights burning eternally in the summer darkness. Modern science tells us these lights burn for a different kind of eternity, as firefly luciferase has become one of the most important tools in medical research, helping scientists track cancer cells, study gene expression, and develop new treatments for diseases. The firefly's glow reminds us that sometimes the most profound innovations come in the smallest packages. As these beetles drift through warm summer air, each flash represents millions of years of evolution, countless chemical reactions perfected through trial and error, and a living technology that surpasses our best attempts at artificial illumination. Their light, so familiar, yet so extraordinary, transforms ordinary evenings into something magical, proving that nature's engineering often exceeds our wildest imagination. Drifting through the twilight waters of the Pacific Northwest, a transparent umbrella pulses with an ethereal green glow. The crystal jelly, Equoria victoria, appears almost ghost-like as it moves through the water column, its bell contracting and expanding in a rhythm as old as the oceans themselves. This seemingly simple creature has revolutionized modern science in ways its graceful movements could never suggest, harboring within its transparent tissues a protein that would eventually illuminate everything from cancer cells to neural pathways. The crystal jelly's bioluminescence begins with a protein called equorin, which produces blue light when it encounters calcium ions. But something magical happens next. This blue light is immediately absorbed by another protein, the now famous green fluorescent protein, or GFP, which re-emits it as a characteristic green glow. This two-step process, called bioluminescence resonance energy transfer, creates the ghostly green aurora that rings the jellyfish's bell like a living crown of light. Scientists first isolated GFP in the 1960s, but it wasn't until decades later that they realized they had discovered one of biology's most powerful tools. Did you know that this protein from a humble jellyfish earned 3 scientists the 2008 Nobel Prize in Chemistry? Researchers discovered they could attach GFP to virtually any protein in living cells, creating a glowing tag that allows them to watch biological processes unfold in real time. Today, modified versions of GFP help scientists track how cancers spread, watch neurons fire in living brains, and observe how proteins move within cells. The crystal jelly produces its light show through a fascinating mechanism. Around the rim of its bell, special light-producing cells, called photocytes, contain both equorin and GFP in precise arrangements. When the jellyfish is disturbed, calcium ions flood into these cells, triggering the equorin to produce its blue flash. The GFP molecules, positioned perfectly to capture this light, instantly convert it to green. The entire process happens in milliseconds, creating waves of green light that ripple around the jellyfish's bell. Research reveals that crystal jellies don't glow constantly, but save their light displays for special occasions. A gentle touch might produce a single pulse of light, while a more aggressive disturbance can trigger a spectacular light show lasting several seconds. Marine biologists believe this serves as a burglar alarm, attracting larger predators to whatever is attacking the jellyfish. It's a clever strategy. If you can't fight off your attacker, call in something bigger that might eat them instead. The green fluorescent protein itself is a marvel of natural engineering. Composed of 238 amino acids folded into a barrel shape, it contains a light-emitting center that forms spontaneously when the protein folds correctly. This self-assembling characteristic makes GFP incredibly useful in research, as scientists need only introduce the gene for GFP into cells, and the protein builds itself without any additional help. The light it produces is so pure and consistent that it has become the gold standard for biological imaging. Crystal jellies have taught us that bioluminescence can be surprisingly sophisticated. Unlike the chemical reaction in fireflies, which requires a constant supply of fresh luciferin, the crystal jelly system is rechargeable. The aquarin protein can bind new calcium ions and produce light again and again, while GFP never gets consumed in the process. This renewable system allows crystal jellies to produce hundreds of flashes throughout their lives without depleting their resources. Scientists recently discovered that crystal jellies can somehow sense the quality of light in their environment and adjust their bioluminescence accordingly. In laboratory studies, jellies exposed to different wavelengths of ambient light showed variations in the intensity and duration of their glowing responses. This suggests these simple creatures possess a more complex relationship with light than previously imagined, perhaps using their bioluminescence not just for defense, but for communication with others of their kind. The impact of the crystal jelly's contribution to science continues to grow. Researchers have now created an entire rainbow of fluorescent proteins inspired by the original GFP, each glowing in a different color. These tools allow scientists to track multiple processes simultaneously, painting living cells in a palette of glowing colors that would make any artist envious. From understanding how memories form to developing new treatments for genetic diseases, the applications seem limitless. In their natural habitat, crystal jellies drift through coastal waters like living auroras, their gentle pulsing creating circles of green light in the darkness. Watching them, it's hard to imagine that these delicate creatures, composed of 95% water, could harbor such powerful biological tools. Yet within their transparent bells lies a reminder that nature's innovations often hide in plain sight, waiting for curious minds to discover their secrets. The crystal jelly's legacy extends far beyond its Pacific home, its green glow now illuminating laboratories around the world. Every time a scientist uses GFP to mark a protein or track a cellular process, they're harnessing millions of years of evolution, packaged in a molecule so elegant that human engineering has yet to improve upon the original design. In the gentle pulse of a jellyfish's light, we find both beauty and utility merged in perfect harmony. Deep within the limestone caves of New Zealand, one of nature's most enchanting light displays unfolds in complete darkness. The ceiling sparkles with thousands of tiny blue-green lights, creating a scene so magical that early explorers believed they had discovered underground stars. These aren't stars, of course, but the larvae of a unique fungus gnat, Arachnocampa luminosa, whose Maori name, Titiwai, means projected over water, perfectly capturing the reflection of their lights in the dark underground streams. These glowworms, which aren't actually worms but fly larvae, have transformed entire cave systems into natural planetariums through one of evolution's most successful hunting strategies. Each larva, no bigger than a matchstick, produces light through a chemical reaction in specialized organs in its tail segment. The biochemistry is similar to other bioluminescent creatures, using luciferin and luciferase, but the way these larvae employ their light is uniquely ingenious and slightly macabre. Scientists studying these caves have documented one of nature's most patient hunting methods. Each glowworm larva constructs a nest of silk on the cave ceiling, then dangles dozens of sticky threads below it, some reaching down as far as 20 inches. These fishing lines, beaded with mucus droplets that sparkle in the larva's light, create a deadly chandelier. Small insects, drawn upward by the glowing light in the darkness, become entangled in the sticky strands. The larva then reels in its catch, recycling both the prey and the silk line in an efficient system that wastes nothing. Did you know that glowworm colonies create their own weather systems within caves? The combined body heat from thousands of larvae can raise the temperature near the ceiling by several degrees, causing air currents that help disperse flying prey throughout the cave. Researchers have measured these microclimates and found that larger colonies actually create circular air patterns that funnel insects toward the waiting fishing lines, showing how these simple larvae collectively engineer their environment. The intensity of a glowworm's light directly correlates with its hunger level. Well-fed larvae glow dimly, conserving energy, while hungry individuals produce a brighter beacon to attract more prey. This creates a fascinating dynamic within the colony, where the brightness of the ceiling actually indicates the overall health of the ecosystem. Tour guides in New Zealand's famous Waitomo caves have learned to read these patterns, knowing that a brilliantly glowing ceiling might mean the colony hasn't eaten well recently. Research reveals that glowworm larvae can maintain their glow continuously for their entire larval stage, which lasts 6 to 12 months, depending on food availability. Unlike fireflies that flash intermittently, these larvae produce steady light for hours on end, an energetically expensive proposition that shows just how crucial the light is to their survival. The chemical reaction is so efficient that the larvae produce virtually no heat, preventing their delicate silk fishing lines from drying out in the constant glow. The transformation from glowing larva to adult fly presents one of nature's strangest contrasts. The adult fungus gnats live only a few days, just long enough to mate and lay eggs. They cannot eat, having no functional mouthparts, and they don't glow at all. Their sole purpose is reproduction, after which they often become entangled in the very fishing lines their offspring will construct. It's a circular system where even the parents become part of the food web that sustains the next generation. Scientists recently discovered that glowworm colonies exhibit a form of light etiquette. In areas where larvae are densely packed, individuals seem to moderate their light output to avoid creating too much competition. This self-regulation prevents the colony from becoming too bright, which might actually deter prey insects that prefer dimmer conditions. It's a delicate balance between attracting enough food and maintaining the dark environment that makes their lights effective. The optical properties of the glowworm's light organ are remarkably sophisticated. The organ contains reflective cells that act like tiny mirrors, directing all the light downward toward potential prey. This biological reflector is so efficient that nearly all the light produced is directed where it's needed, wasting none on the cave ceiling above. Engineers studying these structures have found inspiration for more efficient LED designs, once again showing how nature's solutions often surpass human technology. Cave systems hosting glowworms have become carefully managed ecosystems. Even slight changes in humidity, temperature, or airflow can affect the colonies. Tour operators have learned to limit visitor numbers and control lighting carefully, as too much disturbance can cause the larvae to stop glowing for hours or even days. The delicate balance required to maintain these living light shows reminds us how fragile nature's most beautiful displays can be. In the quiet darkness of New Zealand's caves, these patient hunters continue their ancient practice, each larva a single star in a living constellation. Visitors often report that floating beneath a glowworm colony on an underground river feels like drifting through space, surrounded by galaxies of blue-green stars. It's a profound experience that connects us to the wonder of bioluminescence, showing how life can create beauty even in the deepest darkness. The glowworms remind us that sometimes the most spectacular sights require us to venture into quiet, dark places where nature's light shows bloom in patient silence. Along coastlines around the world, from the Maldives to Puerto Rico, breaking waves sometimes ignite with an ethereal blue fire that has captivated humans for millennia. This living light emerges from microscopic organisms called Dinoflagellates, single-celled algae that transform mechanical energy into chemical light with a sensitivity that surpasses our most advanced senses. These tiny alchemists, measuring just 20 to 200 micrometers across, create one of nature's most breathtaking displays when conditions align perfectly. The species most responsible for these glowing seas is Noctiluca scintillans, commonly known as sea sparkle. Despite being single-celled, these organisms possess a sophisticated bioluminescent system housed within specialized compartments called scintillons. Each scintillon contains the familiar luciferin-luciferase combination, but with a unique twist. The system is triggered by mechanical stress, meaning any disturbance, from a breaking wave to a swimming fish, can set off a cascade of blue light that spreads through the water like liquid lightning. Scientists studying dinoflagellate bioluminescence have uncovered the precise mechanism behind this hair trigger response. When the cell experiences mechanical stress, tiny channels in its membrane open, allowing calcium ions to flood into the scintillons. This calcium influx causes a rapid pH change that activates the luciferase enzyme, producing a flash of light that lasts just 100 milliseconds. The entire process, from stimulus to light, takes less than 20 milliseconds. making it one of the fastest biological responses known to science. Did you know that Dinoflagellates follow a circadian rhythm with their bioluminescence? These microscopic organisms glow much more brightly at night than during the day, even when kept in constant darkness in laboratory conditions. Researchers have discovered that they possess internal biological clocks that regulate their light production, ensuring maximum brightness during nighttime hours when the defensive value of their glow is highest. This rhythmic behavior shows that even single-celled organisms can have sophisticated timing mechanisms. The ecological purpose of dinoflagellate bioluminescence has fascinated marine biologists for decades. The prevailing theory suggests it serves as a burglar alarm, similar to the crystal jellies strategy. When small predators like copepods attempt to eat dinoflagellates, the resulting light show attracts larger fish that prey on the copepods. It's an elegant solution. If you can't defend yourself directly, illuminate your attacker and let something bigger handle the problem. This creates a complex web of interactions where light becomes a currency of survival in the darkness. Research reveals that the intensity of bioluminescent displays varies dramatically with dinoflagellate density and environmental conditions. Spectacular displays typically require concentrations of more than 1,000 cells per liter, though truly memorable events can involve millions of cells per liter. These blooms often follow periods of calm weather and nutrient upwelling when conditions favor rapid reproduction. The result can be miles of coastline, outlined in blue fire, with every wave crest glowing as if lit from within. The biochemistry of dinoflagellate light production shows remarkable efficiency. Unlike many bioluminescent systems that require multiple components, dinoflagellates have evolved a unique luciferin that's among the most unusual in nature. This compound, a tetraperol similar to chlorophyll, allows them to produce light using the same basic molecular framework they use for photosynthesis during the day. It's an elegant example of evolutionary recycling, using similar chemistry for both capturing and producing light. Scientists recently discovered that dinoflagellates can actually exhaust their bioluminescent capacity through repeated stimulation. Each cell contains a finite amount of luciferin that takes hours to regenerate after being consumed. This means that areas of water that have been highly disturbed might temporarily lose their glow, creating dark patches in otherwise luminescent seas. Surfers and swimmers often notice this effect, finding that the water glows brightly on their first pass, but dimmer on subsequent ones. The sensitivity of dinoflagellate bioluminescence has made these organisms valuable as biological sensors. Researchers have developed systems that use dinoflagellate cultures to detect water movement, chemical pollutants, and even biological warfare agents. The organism's light output changes in response to various stresses, providing a living early warning system that's both sensitive and cost-effective. Some military organizations have even studied bioluminescent wakes left by submarines and ships, though dinoflagellates make unreliable spies, glowing for any disturbance regardless of its source. Dinoflagellate blooms capable of producing bioluminescence aren't always benign. Some species produce toxins alongside their light, creating what locals call red tides during the day that transform into blue tides at night. These events can close fisheries and beaches, showing that nature's most beautiful displays sometimes come with hidden dangers. The same organisms that create magical nighttime scenes can also produce some of the most potent natural toxins known to science. The cultural impact of bioluminescent bays has been profound throughout history. Ancient Greek and Roman texts describe glowing seas, often attributing them to divine presence. In Puerto Rico, mosquito bays Dinoflagellates create the brightest bioluminescent bay in the world, with each gallon of water containing up to 700,000 dinoflagellates. Swimming in these waters feels like moving through liquid starlight, with every movement creating trails of blue fire that linger for seconds before fading back to darkness. As our understanding of dinoflagellate bioluminescence grows, so does our appreciation for these microscopic light producers. They remind us that some of nature's most spectacular phenomena emerge from its smallest inhabitants, that single cells can create displays visible from space when they work together. In the gentle lapping of glowing waves on a dark beach, we witness chemistry, physics, and biology converging to create pure magic. In the vast expanse of the open ocean, between the sunlit surface and the eternal darkness below, lives Earth's most abundant vertebrate, though most people have never heard of them. Lanternfish, comprising over 240 species in the family Myctophidae, outnumber all the birds, reptiles, and mammals on our planet combined. These small, silvery fish, rarely longer than 6 inches, undertake the largest migration on Earth every single day, and they do it all by the light of their own bodies. Each lanternfish carries an array of photophores arranged in species-specific patterns along their sides and belly. These light organs, numbering anywhere from a dozen to several hundred depending on the species, create what marine biologists describe as an identification system more reliable than fingerprints. The arrangement is so precise that scientists can identify a lanternfish species in complete darkness just by photographing its light pattern, like reading a glowing barcode in the deep ocean. The daily vertical migration of lanternfish represents one of nature's most coordinated movements. As darkness falls, billions upon billions of these fish rise from depths of 3,000 feet or more toward the surface, following the zooplankton they feed upon. This mass movement is so substantial that early sonar operators mistook it for the ocean floor rising and falling. Scientists now call this phenomenon the deep scattering layer, and it involves such a massive biomass that it can be detected from space using satellite sensors. Did you know that lanternfish use their photophores for a technique called counter-illumination? By producing light on their bellies that matches the dim sunlight filtering down from above, they become nearly invisible to predators looking up from below. This sophisticated camouflage system requires each fish to constantly adjust its light output based on the ambient light conditions, depth, and time of day. It's like wearing an invisibility cloak that needs constant fine-tuning, yet lanternfish manage this complex task instinctively. Research reveals that different species of lanternfish partition the ocean's twilight zone through their light patterns and behaviors. Some species migrate to within 100 feet of the surface, while others stop at 500 feet. Their photophore patterns help them recognize their own kind in the darkness, preventing energy-wasting competition between species. This vertical segregation, mediated by light, allows hundreds of species to coexist in what appears to be a featureless water column. The biochemistry of lanternfish bioluminescence shows remarkable adaptations to their mobile lifestyle. Unlike many deep-sea fish that rely on bacterial symbionts for light, lanternfish produce their own luciferin and luciferase. This self-sufficiency allows them to maintain consistent light production throughout their extensive daily journeys. Their photophores can produce light continuously for hours, essential for maintaining camouflage during their long migrations through changing light conditions. Scientists recently discovered that lanternfish schools coordinate their light displays in ways we're only beginning to understand. High-speed cameras have captured waves of light passing through schools, suggesting some form of communication beyond simple species recognition. These coordinated displays might help maintain school cohesion in the darkness, coordinate feeding, or even confuse predators. The patterns are reminiscent of starling murmurations, but conducted with light instead of movement. The ecological importance of lanternfish extends far beyond their impressive numbers. These small fish form a crucial link in ocean food webs, converting tiny zooplankton into protein that feeds everything from squids to whales. Sperm whales dive thousands of feet specifically to feast on lanternfish schools, while dolphins and seabirds wait for them to approach the surface at night. The nightly migration of lanternfish represents one of the largest movements of biomass on Earth, transferring nutrients from the depths to the surface in a cycle that helps drive ocean productivity. The pressure changes lanternfish experience during their daily migrations would kill most fish. Rising from 3,000 feet to near the surface involves a pressure change of about 100 atmospheres. Lanternfish accomplish this using specialized swim bladders that can rapidly adjust to pressure changes, accompanied by photophores that maintain consistent light output despite the physical stress. It's equivalent to climbing Mount Everest and back every single day, all while keeping hundreds of tiny lights perfectly calibrated. Recent studies using underwater robots have revealed that lanternfish schools create what researchers call false bottoms in the ocean. Their combined bodies and swim bladders reflect sonar so strongly that they appear as solid surfaces on instruments. During World War II, these phantom sea floors confused submarine operators who couldn't understand why the ocean depth seemed to change at night. We now know they were detecting one of nature's greatest light shows, performed by Earth's most numerous vertebrates. The metabolic cost of producing light while undertaking such extensive migrations has puzzled scientists. Lanternfish seem to have solved this energy equation through exceptional efficiency in both swimming and light production. Their bodies are perfectly streamlined for vertical movement, while their photophores convert chemical energy to light with minimal waste. This efficiency allows them to maintain their nomadic lifestyle while still producing enough light to stay camouflaged and communicate with their schoolmates. As we peer into the ocean's twilight zone, lanternfish remind us that the most numerous animals on our planet are often the least visible. Their nightly ballet, performed in darkness and choreographed by light, represents one of nature's most successful strategies. Each tiny fish, glowing softly as it rises through the darkness, is part of a movement so vast it changes the very structure of the ocean every night and day. You drift slowly down through mellow midnight water, settling into the soft, silty floor of the continental shelf. Around you, nothing stirs except a measured sway, as if the sea itself is breathing in long, unhurried intervals. Then a glow appears, faint at first, like the memory of fireflies, and you realize you have entered a living meadow. Hundreds of pale pink quills stand rooted in the sand, each topped with delicate feathery branches that curve upstream like tiny trees bowing to a gentle wind. These are sea pens, colonial relatives of corals and anemones, and tonight they have chosen to write their quiet story in pulses of light. The light begins when a passing current brushes one of the polyps. Almost shyly, a small green spark blooms at the point of contact, then travels up the central stalk in a slow ripple. A neighboring sea pen senses the glow and answers with a flare of its own. And soon, the meadow is speaking in luminous whispers, wave after wave of cool emerald radiance sliding through the forest of quills. Scientists have measured this traveling glow at about 2 centimeters per second, slow enough that you can follow each pulse from base to tip like a line of lanterns carried up a winding staircase. Within minutes, the entire colony is alive with light, a silent conversation conducted in photon syllables. Sea pens belong to the order Penetrulacia, named for the quill-like rachis that anchors the colony. Unlike reef-building corals cemented to rock, a pen inserts its root-like peduncle deep into soft sediment, inflates with seawater, and stands upright like a miniature flagpole. Each branch along the rachis holds dozens of feeding polyps, tiny 8-tentacled creatures that unfurl to catch plankton drifting by. During the day, most sea pens withdraw, deflating until only a pale nub shows at the sediment surface. But at night, they reinflate and extend their polyps, and in some species, bioluminescence stands ready as a shield against intrusion. The chemistry behind the glow is as elegant as it is efficient. A luciferin molecule meets the enzyme luciferase in the presence of oxygen and calcium ions, releasing a cool green photon without producing measurable heat. In laboratory studies, researchers have found that sea pens can flash repeatedly for hours without apparent fatigue, suggesting a generous supply of chemical fuel. Yet the glow is never wasted. It is reserved for moments when the colony is touched, disturbed, or, for reasons we still do not fully understand, when a dense nocturnal current passes through, as though the meadow feels compelled to greet the water itself. Did you know that the first written record of sea pen light dates back to 18th century sailors who reported luminous quills sprouting from the ocean floor? They believed the glow to be the work of underwater spirits, for no flame they knew could burn beneath the waves. Modern submersible cameras confirm those tales. Entire slopes can sparkle when trawling gear drags across the sediment, a defensive response that may startle fish or crustaceans long enough for polyps to retract. In nature, the trigger is gentler, a grazing starfish, a wandering isopod, but the principle stands: better to dazzle than to be devoured. Each sea pen colony is, in truth, a single animal made of many clones. The central stalk is a modified polyp that devotes itself to support, while the lateral polyps handle feeding and, when the season is right, reproduction. Scientists recently discovered that some species synchronize the release of gametes with lunar cycles, their internal clocks guided perhaps by minute changes in nocturnal blue light filtering from above. On those spawning nights, the meadow may glow more frequently, the pulses traveling faster, as though the colony is rehearsing its own celestial alignment before sending tiny larvae into the currents. The meadow sways again, and you notice the rhythm of the polyp's tentacles. Each frond moves in slow circular gestures, stirring the boundary layer of still water that hugs the colony. By breaking this layer, the sea pens increase the flow of oxygen and plankton across their surfaces, a passive feeding strategy refined by millions of quiet nights. When a rich plankton patch arrives, the polyps capture copepods, diatoms, and microcrustaceans with mucus-lined tentacles. Beneath that serene exterior, tiny stomachs busily digest, transforming drifting life into energy that will fuel tomorrow's glow. Research divers have measured colonies more than 1 meter tall, yet most individuals you pass tonight stand scarcely 20 centimeters. Size, it seems, is a balance between the stability offered by deep anchoring and the drag imposed by stronger currents higher in the water column. Some polyps at the tips of tall pens specialize in reproduction, while those closer to the base feed. This vertical zoning makes the colony more efficient, allowing each polyp to focus on its strongest contribution, an arrangement not unlike the specialization in a beehive or a human society. Zoologists still debate whether sea pen bioluminescence serves primarily as defense, communication, or both. Laboratory experiments show that repeated prodding by shrimp triggers longer flashes, suggesting a startle function. Yet in situ observations reveal synchronous waves of light that travel across dozens of colonies without any apparent predator, implying a form of meadow-wide signaling, perhaps a nightly check-in that says, "I am here and the water is good." Because the glow costs energy, it must confer some advantage, and the answer may lie in a combination of deterrence and community coordination. A gentle downdraft lifts a swirl of sediment, momentarily shadowing the scene. For an instant, the meadow dims, polyps withdrawing as grains of sand settle on their delicate tentacles. Then, one by one, they re-extend, brushing off the debris, reopening to the current. You can almost hear their patient sigh. Life on the seafloor means adapting to each whisper of mud, each eddy of darkness, and responding not with panic, but with a measured glow. If you approach slowly and lay a fingertip, or in this imagined drift, a single curious thought, against the rachis, a new pulse blossoms, emerald at the point of contact. It climbs the stalk like a slow exhalation, reaching the farthest polyp before fading back to the gentle background glow. In that moment, you sense the time scale of these beings, unhurried, deliberate, their bioluminescent vocabulary spoken in sentences a full minute long. It is language stretched out for the patient listener, perfect for a mind ready to relax into slower rhythms. Some night, remote camera footage shows a squat lobster grasping the base of a sea pen, attempting to nip at its tissue. Instantly, the pen retracts half its length into the sediment while flashing furiously, a living flare signaling distress. Neighboring pens echo the flash and duck as well, the meadow folding into itself like umbrellas in sudden rain. Minutes later, when the lobster scuttles off, the quills rise again, polyps reopening as if nothing happened. The glow subsides to soft candlelight, and the sea resumes its breath. To study sea pens, scientists deploy sediment cores, gentle suction samplers, and low-light cameras. Back on deck, researchers keep the pens in chilled seawater tanks, dim red lamps preserving their night vision. A small brush, simulating a predator, can trigger light for spectral analysis. Most species radiate between 490 and 515 nanometers, right in the range seawater transmits best. That wavelength paints the meadow green to the human eye, though down here, surrounded by blue-filtered starlight, the glow feels almost white. You begin to ascend, leaving the luminous garden below. From above, the meadow is barely visible, its quills thin as pins against the wide plain of silt. Yet you know that should a curious fish or distant submarine pass, the meadow can become a beacon, a slow-motion aurora grounded in sand. It is an ecosystem that communicates through patience and light, an invitation to slow down, to notice every ripple of water and pulse of glow. Long after you have drifted upward into darker water, a final wave of light sweeps the field. Perhaps a goodnight ripple, perhaps a message only other pens can decode. Either way, the sea pens carry on, writing their luminous sentences across the sea floor, chapter after chapter, night after tranquil night. Drifting through oceans worldwide, from tropical shallows to polar depths, swim creatures that create living rainbows without producing a single photon of their own light. Comb jellies, or ctenophores, are among Earth's most ancient animals, with fossils dating back over 500 million years. These translucent, walnut-shaped creatures generate one of nature's most mesmerizing light displays through pure physics, proving that not all that glows in the ocean relies on biochemistry. The secret to the comb jelly's rainbow lies in 8 rows of cilia, microscopic hair, like structures arranged in bands called tenaes, from which they derive their scientific name. Each band contains thousands of cilia that beat in coordinated waves, propelling the animal through the water with surprising grace. As these cilia move, they act like living diffraction gratings, splitting white light into its component colors. The result is ribbons of rainbow light that ripple along the creature's body, creating an effect so beautiful it seems almost supernatural. Scientists studying comb jellies have discovered that this light show is purely optical, requiring no energy expenditure beyond the normal ciliary movement used for swimming. When sunlight or artificial light hits the moving cilia at just the right angle, it diffracts into spectacular rainbow patterns. The spacing between cilia, typically about 1 micrometer, is perfectly sized to create interference patterns with visible light. It's essentially the same physics that creates rainbows on soap bubbles or oil slicks, but executed by living tissue in constant motion. Did you know that comb jellies were once mistaken for bioluminescent creatures by early marine biologists? The confusion is understandable, as many species do possess true bioluminescence in addition to their rainbow cilia. However, the rainbow effect can be observed even in dead specimens when their cilia are artificially moved, proving it's a purely physical phenomenon. This dual ability to create light through both chemistry and physics makes comb jellies unique among marine organisms. The coordination required to produce smooth rainbow waves along their bodies reveals sophisticated neuromuscular control. Unlike jellyfish, their distant relatives, comb jellies possess a simple nerve net that coordinates ciliary beating with remarkable precision. High-speed cameras have captured the metachronal waves where each cilium beats slightly after its neighbor, creating the smooth, flowing motion that generates the rainbow effect. This coordination happens automatically, requiring no conscious control, like a heart beating or lungs breathing. Research reveals that different species of comb jellies create different rainbow patterns based on their ciliary arrangements and body shapes. The common sea walnut, Mnemiopsis leedi, produces broad rainbow bands that spiral around its body as it swims. The Venus girdle, Kestum veneris, a ribbon-shaped comb jelly that can grow several feet long, creates rainbow waves that undulate along its entire length like aurora borealis in miniature. Each species has evolved its own variation on the rainbow theme, adapted to its particular lifestyle and habitat. The evolutionary history of comb jellies has recently revolutionized our understanding of animal evolution. Genetic studies suggest these creatures might be the sister group to all other animals, possibly even predating sponges. This means the sophisticated ciliary system that creates their rainbows might represent one of nature's earliest experiments in coordinated cellular movement. The rainbow effect, therefore, could be one of the oldest light displays in the animal kingdom, predating true bioluminescence by millions of years. Scientists recently discovered that comb jellies can modulate their rainbow displays through subtle changes in swimming patterns. Slower beats create more distinct color bands, while rapid swimming blurs the colors into a shimmering halo. Some species appear to use these variations for communication, creating specific patterns when encountering others of their kind. This suggests that even a purely physical light display can be behaviorally controlled and potentially carry information. The transparency of comb jellies adds another dimension to their light displays. Their bodies, composed of 99% water, are nearly invisible except for the rainbow cilia and, in some species, bioluminescent photocytes. This combination creates creatures that seem to be made entirely of light and water, drifting through the ocean like living prisms. Photographers struggle to capture their ethereal beauty as the rainbow effect changes dramatically with viewing angle and lighting conditions. The ecological role of the rainbow display remains somewhat mysterious. Unlike bioluminescence, which clearly serves defensive or offensive purposes, the rainbow effect seems to be largely an accident of physics. However, some researchers hypothesize that the constantly shifting rainbow patterns might confuse predators or make it difficult to judge the comb jelly's exact position in the water. The display might also help comb jellies recognize each other in the vast ocean, using the specific pattern of rainbow waves as a species identifier. In the sunlit waters where comb jellies drift, they remind us that nature creates beauty through multiple pathways. Their rainbow displays, requiring no complex chemistry or energy expenditure, show that sometimes the most spectacular effects arise from simple physical principles applied to living structures. Each comb jelly, pulsing through the water with its crown of rainbows, demonstrates that the marriage of physics and biology can produce magic just as profound as any biochemical light show. In the shallow coastal waters of Hawaii and the Indo-Pacific, a creature no bigger than a golf ball has perfected one of nature's most adorable survival strategies. The Hawaiian bobtail squid, Euprymna scolopies, looks like it was designed by a committee determined to create the ocean's most endearing cephalopod. With its rounded mantle, stubby arms, and enormous eyes, this tiny squid has become a superstar of symbiosis research, harboring a secret that transforms it into a living invisibility cloak each night. Unlike their larger cousins, who jet through open waters, bobtail squids spend their days buried in sand with only their eyes protruding, waiting for darkness to emerge and hunt. But here's where their story becomes extraordinary. Within a specialized light organ on the squid's underside lives a colony of bioluminescent bacteria, Vibrio fischeri, that provides the squid with a personalized nightlight perfectly calibrated to match the moonlight filtering down through the water. Scientists studying this partnership have uncovered one of nature's most precise examples of counter-illumination camouflage. The bobtail squid's light organ isn't just a simple pocket of glowing bacteria, but a sophisticated optical instrument, complete with reflectors, lenses, and filters. The squid can control the intensity and direction of the bacterial light, creating a glow on its underside that exactly matches the moonlight above. To predators looking up from below, the squid simply vanishes, its silhouette erased by its bacterial accomplices. Did you know that baby bobtail squids are born without their glowing partners? Within hours of hatching, the tiny squids must recruit the right bacteria from the millions of microbes in the surrounding seawater. The squid's light organ produces a special cocktail of chemicals that attracts only Vibrio fischeri while repelling or killing other bacteria. It's like posting a help wanted ad that only one species can read. Once the right bacteria arrive, the squid's immune system, which would normally attack foreign invaders, welcomes them in and helps them establish their new home. The daily rhythm of this partnership reveals remarkable coordination between host and symbiont. Each dawn, the bobtail squid expels about 95% of its bacterial partners, releasing them into the ocean. The remaining bacteria spend the daylight hours multiplying, repopulating the light organ to full capacity by nightfall. This daily purge serves multiple purposes. It prevents the bacteria from overgrowing, ensures a fresh, healthy population each night, and seeds the ocean with bacteria for the next generation of squids to recruit. Research reveals that the bobtail squid can fine-tune its bacterial light show with extraordinary precision. The light organ contains specialized tissues that can block or redirect light, allowing the squid to adjust its glow based on cloud cover, moon phase, and water clarity. Scientists have observed squids dimming their bacteria on overcast nights and brightening them when the moon is full. This isn't conscious control, but rather an automatic response mediated by photoreceptors that monitor the downwelling light and adjust the bacterial glow accordingly. The biochemistry of Vibrio fischeri's bioluminescence has become a model system for understanding bacterial communication. These bacteria only produce light when they reach a certain population density, using a process called quorum sensing to count their neighbors. Each bacterium releases signaling molecules, and when enough accumulate, it triggers the genes for light production. This ensures the bacteria don't waste energy producing light when too few are present to create a useful glow. It's cellular democracy in action, with bacteria voting on when to illuminate their squid host. Scientists recently discovered that the relationship between bobtail squids and their bacteria influences the squid's development in profound ways. Squids raised without their bacterial partners develop abnormal light organs and altered gene expression patterns. The bacteria actually help shape the organ they inhabit, triggering developmental changes that optimize the structure for light production and distribution. It's a remarkable example of how symbiotic partners can influence each other's biology at the most fundamental levels. The ecological importance of this partnership extends beyond the individual squid. The daily expulsion of bacteria creates pulses of bioluminescent microbes in coastal waters, contributing to the background glow often observed in tropical seas. These free-living bacteria can survive for days in the ocean, ready to colonize the next generation of baby squids. Some researchers suggest this system helps maintain the genetic diversity of both partners, as squids sample from a vast oceanic pool of bacteria rather than simply inheriting them from their parents. Laboratory studies have revealed that bobtail squids can be quite particular about their bacterial partners. When offered different strains of Vibrio fischeri, squids show preferences for certain genetic variants that produce optimal light characteristics. This selectivity suggests an ongoing evolutionary dance between host and symbiont, with each generation fine-tuning the partnership for maximum effectiveness. The result is a relationship so integrated that neither partner functions optimally without the other. The bobtail squid's approach to predator avoidance through bacterial partnership has inspired biomimicry research. Engineers studying counter-illumination camouflage for underwater vehicles have drawn insights from the squid's light organ structure. The combination of biological light production with sophisticated optical control represents a solution more elegant than any current technology. Some researchers envision future submarines coated with bacterial films that could provide adaptive camouflage without the need for complex electronics. In the gentle waters where bobtail squids hunt, they demonstrate that sometimes the best defense is to become one with the light itself. These tiny cephalopods, glowing softly with the help of their bacterial partners, remind us that cooperation can emerge at any scale, from the microscopic to the magnificent. Each night, as they emerge from the sand with their living lanterns perfectly calibrated, they perform a vanishing act that transforms the ordinary moonlight into a cloak of invisibility. In the warm Caribbean waters around Jamaica, Belize, and other tropical coastlines, one of nature's most spectacular yet intimate light shows unfolds just after sunset. Tiny crustaceans called ostracods, no larger than tomato seeds, emerge from their daytime hiding places to perform a bioluminescent mating display that rivals any fireworks celebration. These seed shrimp, as they're commonly known, have transformed the serious business of reproduction into one of the ocean's most enchanting spectacles. The species Vagula hilgendorfi, along with several Caribbean relatives, produces one of the most complex bioluminescent displays in the animal kingdom. Males release precisely timed pulses of glowing chemicals into the water, creating trails of blue light that hang in the water column like sapphire pearls. Each species has its own unique pattern, some creating vertical lines, others horizontal spirals, and still others producing elaborate three-dimensional light sculptures that persist for several seconds before fading into darkness. Scientists studying ostracod displays have discovered that these light shows represent a form of competitive advertising unique in the natural world. Unlike fireflies or other bioluminescent creatures that produce light within their bodies, male ostracods mix their luciferin and luciferase outside their bodies in the open water. This external mixing creates clouds of chemical light that can be far larger and brighter than anything their tiny bodies could produce internally. It's essentially a way of creating a billboard-sized advertisement from a creature smaller than a grain of rice. Did you know that female ostracods judge potential mates based on the precision and brightness of their displays? Researchers have found that successful males produce pulses at exactly the right intervals, creating evenly spaced light pearls that demonstrate their fitness and genetic quality. Males that produce irregular patterns or dim lights are ignored, their chemical investments wasted in the darkness. This selective pressure has driven the evolution of increasingly sophisticated and precise displays over millions of years. The biochemistry of ostracod bioluminescence shows remarkable adaptations for their unique external light production. These crustaceans store their luciferin in specialized glands, keeping it separate from the luciferase until the moment of display. When ready to signal, they squirt both chemicals from different glands simultaneously, allowing them to mix in the seawater. The reaction is instantaneous and brilliant, producing some of the brightest bioluminescence relative to body size found anywhere in nature. Research reveals that ostracod mating displays follow strict environmental cues. The performances begin precisely at nautical twilight, when the sun is 12 degrees below the horizon, and continue for exactly 1 to 2 hours. Cloud cover, moon phase, and water temperature all influence the intensity and duration of displays. On perfect nights, with calm, warm water and no moon, thousands of males might perform simultaneously, creating what observers describe as underwater meteor showers. The diversity of ostracod light patterns has provided scientists with insights into speciation and evolution. In the Caribbean alone, researchers have identified over a dozen species based primarily on their unique light displays. Each pattern is like a species-specific love song written in light, preventing hybridization between closely related species. Some create simple upward streaks, while others produce complex spirals that would challenge a computer programmer to replicate. This diversity arose through sexual selection, as females drove the evolution of ever more elaborate and distinctive displays. Scientists recently discovered that ostracods can adjust their displays based on competition. When multiple males display in the same area, they space their performances to avoid interference, creating a coordinated spectacle where each male gets his moment in the spotlight. This cooperation among competitors is unusual in nature and suggests a level of social awareness surprising in such tiny creatures. The result is a harmonious light show where individual interests align to create collective beauty. The ecological impact of ostracod displays extends beyond reproduction. The bright pulses of light attract small fish which feed on the ostracods, which in turn attracts larger predators. For a brief period each night, these tiny crustaceans become the center of a feeding web, their quest for love inadvertently feeding the reef community. It's a reminder that in nature, even the most intimate moments connect to larger ecological patterns. The study of ostracod bioluminescence has practical applications in biotechnology and environmental monitoring. Their external light production system, requiring only the mixing of two chemicals, has inspired development of new emergency lighting systems and biological sensors. The precise timing of their displays also makes them excellent indicators of environmental health, as pollution or temperature changes disrupt their carefully choreographed performances. Cultural connections to ostracod displays run deep in coastal communities. In Japan, where Vagula hilgendorfi is called umi hotaru or sea firefly, their dried bodies were once ground into powder and used as emergency light sources by soldiers and fishermen. A pinch of powdered ostracods mixed with water would glow brightly enough to read by, creating light from death that mirrors the creature's own external chemistry. Modern tourists flock to witness living displays, supporting local economies through bioluminescence tourism. As our journey through nature's living lights draws to a close, the ostracods offer a fitting finale. Their brief, brilliant displays remind us that bioluminescence serves life's most fundamental purpose, the continuation of species through successful reproduction. Yet in fulfilling this basic need, they create beauty that transcends function, turning the practical matter of finding mates into art painted with light on the ocean's dark canvas. Along the rocky shores of Australia and New Zealand lives a small marine snail with an illuminating secret that scientists discovered only recently. The Clusterwink snail, Hinea brasiliana, appears unremarkable at first glance, just another small gastropod in a plain brown shell barely an inch across. But when threatened, this humble mollusk transforms into a living beacon, using its shell as a natural fiber optic cable to amplify and broadcast bioluminescent light in a way found nowhere else in nature. The Clusterwink snail's light-producing cells are located in its soft body, particularly concentrated in the mantle tissue that lines the shell's interior. When disturbed, these cells produce pulses of blue-green light through the standard luciferin-luciferase reaction. But here's where things get interesting. Instead of the light being visible only where the snail's soft body emerges from the shell, the entire shell glows like a tiny lampshade, broadcasting light in all directions. Scientists studying this phenomenon discovered that the Clusterwink's shell acts as a sophisticated light diffuser. The shell's crystalline structure, composed of calcium carbonate arranged in precise patterns, channels bioluminescent light throughout its surface. Light produced inside the shell undergoes total internal reflection, bouncing through the shell material before emerging evenly across the entire surface. It's as if the snail invented biological fiber optics millions of years before humans developed the technology. Did you know that the Clusterwink snail's shell amplification increases the visibility of its bioluminescence by a factor of 10? Researchers measuring light output found that the shell-diffused glow can be seen from much greater distances than the direct light from the snail's body alone. This amplification transforms what would be a modest warning flash into a beacon visible throughout the snail's rocky habitat. The effect is so pronounced that clusterwinks can illuminate entire tide pools with their defensive displays. The ecological function of this amplified light show appears to be a sophisticated burglar alarm. When crabs or other predators attack, the snail's bright, whole-shell display attracts the attention of the predator's own enemies. Fish that feed on crabs are drawn to the commotion, often interrupting the attack on the snail. It's a clever strategy. If you can't fight back directly, turn your shell into a spotlight that calls for backup. The amplification ensures the signal reaches as many potential defenders as possible. Research reveals that clusterwink snails can produce different patterns of light depending on the type of threat they perceive. A gentle touch might elicit a single pulse of light, while a more aggressive disturbance triggers rapid flashing that can continue for several minutes. The snails seem capable of modulating both the intensity and frequency of their displays, suggesting a more complex defensive strategy than simple reflex responses. This behavioral flexibility allows them to calibrate their energy expenditure to match the level of danger. The physics of light transmission through the clusterwink's shell has fascinated materials scientists. The shell's microstructure contains aragonite crystals arranged in layers that create natural waveguides for light. These biological optical fibers are remarkably efficient, losing very little light intensity as it travels through the shell material. Some researchers have suggested that understanding this natural system could lead to improvements in fiber optic technology or the development of new bioluminescent materials. Scientists recently discovered that juvenile clusterwink snails begin producing bioluminescence before their shells are fully developed. In these young snails, the light display is much more localized, visible only near the shell opening. As the shell grows and its crystalline structure becomes more complex, the light-diffusing properties improve dramatically. This developmental progression suggests that the shell's optical properties are under strong evolutionary selection, with better light diffusion providing increased survival advantages. The clusterwink's bioluminescent system shows remarkable efficiency in its use of resources. Unlike some marine organisms that produce light continuously or for extended periods, clusterwinks flash only when necessary, conserving their luciferin reserves. Each flash typically lasts just a fraction of a second, but the shell's amplification ensures maximum impact from minimal chemical investment. This efficiency allows clusterwinks to maintain their defensive capability even in nutrient-poor environments where energy conservation is crucial. Laboratory studies have revealed that clusterwink snails can detect and respond to the bioluminescence of their neighbors. When one snail in a group begins flashing, others nearby often join in, creating a coordinated light display across the rocky habitat. This contagious flashing might serve to confuse predators or amplify the burglar alarm effect, making it even more likely that higher-level predators will investigate the disturbance. It's a community defense system mediated by light. The unique properties of the Clusterwink's shell have inspired biomimicry research in several fields. Architects interested in natural lighting have studied how the shell diffuses light so evenly, hoping to develop building materials that could distribute artificial or natural light more efficiently. The shell's ability to spread point source light across a broad surface area without significant loss has applications in display, technology, and illumination design. Recent investigations into the Clusterwink's habitat preferences have shown that these snails are most abundant in areas with good water flow and plenty of crevices. The combination of hiding places and current ensures that their bioluminescent signals can travel effectively through the water while providing escape routes when predators approach. This habitat selection demonstrates how the evolution of their unique defense mechanism has influenced their entire ecological niche. The biochemistry of Clusterwink bioluminescence, while following the basic luciferin-luciferase pattern, shows some interesting adaptations to the intertidal lifestyle. The snails can produce light across a range of temperatures and salinities, reflecting the variable conditions of their rocky shore habitat. Their light-producing cells remain functional even during low tide exposure, though the snails rarely flash when out of water, possibly to avoid attracting terrestrial predators. Scientists studying Clusterwink populations have noticed variations in shell thickness and translucency between different locations. Snails from areas with higher predation pressure tend to have shells that diffuse light more effectively, suggesting ongoing evolution of this defensive trait. This geographic variation provides a natural laboratory for understanding how bioluminescent systems and their associated structures evolve in response to ecological pressures. The discovery of the Clusterwink's shell amplification has led researchers to re-examine other bioluminescent mollusks for similar properties. While no other species shows quite the same degree of whole-shell illumination, several related snails exhibit partial light diffusion through their shells. This suggests that the Clusterwink system might represent the most advanced example of a more widespread phenomenon, pushing the boundaries of how organisms can manipulate and amplify their bioluminescent signals. In the tide pools, where Clusterwink snails make their homes, they've created one of nature's most elegant solutions to the problem of being small and vulnerable. By transforming their protective shells into light-amplifying beacons, they've turned a passive defense into an active one. Each flash from a threatened Clusterwink demonstrates that innovation in nature often comes from combining existing elements in new ways. Their shells, evolved for protection, became optical instruments that broadcast danger signals across the reef. As we conclude our journey through nature's living lights, the Clusterwink snail offers a perfect final example of bioluminescent innovation. These small snails remind us that even the humblest creatures can harbor extraordinary capabilities. Their ability to turn a simple shell into a fiber-optic beacon shows that nature's solutions often exceed our technological imagination. In the gentle glow of a Clusterwink's amplified light, we see millions of years of evolution crafting an elegant answer to an ancient problem: how to be noticed when it matters most, using nothing more than the shell on your back and the light within. As the last, gentle pulse of light fades from the Clusterwink's amplified shell, we find ourselves at the peaceful end of our bioluminescent journey. From the synchronized flashes of summer fireflies to the microscopic sparkle of Dinoflagellates in breaking waves, each creature we've encountered tonight has revealed a different chapter in nature's book of living light. These gentle illuminators remind us that even in the deepest darkness, life finds extraordinary ways to create its own radiance. The crystal jellies drifting through twilight waters, their green glow revolutionizing medical science, and the patient glowworm larvae transforming New Zealand caves into underground galaxies. Each represents millions of years of evolution, crafting light from chemistry. The vast schools of lanternfish, rising through the darkness in Earth's greatest migration, their bellies glowing with precise counter-illumination, show us that sometimes the most numerous creatures are also the most invisible. Railroad worms, with their jewel-colored headlights, hunting with red light invisible to their prey, and comb jellies creating rainbows through pure physics rather than chemistry, demonstrate nature's diverse approaches to biological illumination. In warm waters, tiny ostracods perform their nightly ballet of love, painting the sea with trails of blue light, while bobtail squids vanish into moonbeams with the help of their bacterial partners. And in tide pools, Clusterwink snails transform their shells into fiber-optic beacons, proving that innovation can emerge from the humblest of creatures. As you drift towards sleep tonight, perhaps you'll carry with you images of these gentle lights, the patient glow of creatures managing their own illumination, the soft pulse of bioluminescence serving life's essential needs. In a universe vast and often dark, these small beacons remind us that life itself creates wonder in the most unexpected ways. May your dreams be softly lit by nature's living stars, glowing peacefully in oceans, forests, and shores around our remarkable planet. That's all for today's journey through nature's wonders. Sweet dreams.
Sleep, sweet, drift away. Tomorrow brings a brand new day. Dream, dream, to sail so far. You're my precious little star. Shh.